So let's move on to the second video, which is on origin of life. So far we have only discussed about different evolutionary theories and exactly how evolution takes place, different kinds of thoughts regarding that. But how life exactly originated on Earth, that was not yet discussed, or no one ever said anything about that. So let us now move into the theories of regarding origin of life. So how exactly life originated and the theories surrounding it. So the first theory which is now discarded is this theory of special creation given by Father Suarez. So he actually gave this theory that there was this some supernatural powers, and the like we have all heard of Adam and Eve and how they were created by God. So this theory was given by Father Suarez, and of course it was not considered as a correct theory, or there was not any truth behind it. So next thing that we got was the panspermia theory or the cosmozoic theory, where this theory actually suggested that the seed of life exists all over the universe and it can be propagated through space. So suppose there is a meteor crash or a meteorite hitting on the earth, and then the life started originating. So it contains some organisms that survive in extreme conditions, and then they come to earth and in the form of resistant spores. So here also you see a comet arriving on the earth, and this comet has a bacterial life form which is transmitted through space, and therefore when it hits the earth has this kind of organism and it propagates. So that's how the origin of life was there. So these theories are now actually discarded, and let's move on to the next theory, which is Oparin-Haldane theory. So JBS Haldane and Alexander Oparin, they in 1920s they independently set forth the ideas that how exactly the life on earth was. So how exactly the life originated on earth. So pre-life earth, like before life originated, in that earth, how was the environment? They first tried to understand that, and based on that they tried to understand what can be the chemical reactions in that kind of atmosphere, in that kind of environment, so how it can lead to the formation of a cell. So this is also known as Haldane's soup. The simple molecules contained in the soup, they build the complex molecules which are known as the building blocks of life. And from here they form the aggregates, which are also called quasarvates and microspheres. I will go into that later in details. And then you have the protocells. From these aggregates, the protocells or the primitive cells form, and the protocells mixed with nucleic acid, when there is the self-replicating power, then we can call it a cell. So organic molecules can be formed from inorganic molecules in the presence of an external energy source. So this theory was given by Haldane and Oparin, that a primitive earth, primitive atmosphere was free of oxygen. So in this anaerobic condition, how can this chemical reactions take place? So they started doing experiments, and they got very clear results that first life forms must have appeared in the warm primitive ocean, which were actually where actually you can all the simple molecules are present in the water, and therefore it is known as a soup. So this is known as the primordial soup theory, because this John Haldane and Alexander Oparin, they both suggested that the early atmosphere contained these gases like ammonia, CO2, water vapor, etc. And at high temperatures, these gases can form organic compounds. So when this earth cooled down and the water vapor also condensed, so these organic compounds can get collected in the lakes and oceans and also the water bodies. They get solubilized in that. Over time, now these compounds, they can form complex compounds by means of chemical reactions, and also they can get energy from lightning or ultraviolet radiation or any kind of natural energy sources. So from here it was assumed that in this soup, or in this primordial soup, these reactions take place, and therefore the from these complex molecules, actually the protocells or the primitive cells, they can be formed. So there was another theory called the abiogenesis theory, in which suggested that the origin of life was based on certain abiogenic or non-living factors. So gases would assemble into organic compounds, and which would further assemble into complex molecules such as proteins. So this theory explains that there is spontaneous generation of theory of spontaneous generation was given by Aristotle first, which means that life can arise from non-living matter. So we now know that this is practically impossible, because he saw that when you keep meat in the open, then maggots start coming there, and then the maggots form. So he assumed that the meat can give rise to maggots, but it is not true, because we know that the meat must have some kind of they have laid eggs over there, and therefore the maggots have come. So life cannot magically appear. So this idea of spontaneous generation is basically wrong idea. Also, he suggested that mud produces fish, like they have seen mud skippers, so they assume that from the mud only this fish has originated. Also, grain produces mice, which is also not true. Whenever you keep a food source which is unrestricted, then the mice or the pests will obviously come and attack it. So from here, this theory of spontaneous generation was also discarded. So moving on with this Operin-Haldan theory, or the primordial soup theory, we can say that this primitive cell can be formed by the formation of complex sugar from simple sugars, lipid membrane from fatty acids, proteins from amino acids, DNA, RNA from nucleotides. And these inorganic compounds, when they synthesize organic monomers, it forms the polymers, and therefore the formation of pre-cells or protocells, and then we get a membrane enclosed compartment, and also inside that we can get self-replicating molecules or nucleotides. Then we can call it a true cell. So this is the way this theory was proposed, and therefore Miller and Urey performed this experiment, this very famous experiment, to support this Operin-Haldan theory. So this experiment showed that building blocks of life are very simple compounds, and therefore they can build complex compounds, which are the main origin of life. So in this experiment, they have kept this flask with these gases, methane, ammonia, and water vapor. So boiling chamber, from here the water is boiling, and the water vapor is coming through this pipe here. So in this condition, these reactions can take place step by step, and therefore when this is passed on through this, there is a condenser which cools down these gases and solubilizes them. So in this solution, you can get the organic molecules. So first organic molecules appear over here. So when you give an electric spark, the energy is generated over here, and this energy causes the chemical reactions to take place, and this cool water condenses the water vapor, and the soluble molecules present over here are dissolved, and therefore it comes out as this. The organic molecules are collected here. So CO2 breaks down into CO and nascent oxygen, and this oxygen reacts with methane to form CH2O and H2O, and then carbon monoxide reacts with ammonia to form hydrogen cyanide and water, and then methane reacts with ammonia to form hydrogen cyanide and hydrogen, and these compounds react with each other to form amino acids. So this reaction takes place in this lab setup, and the environment that is created here, which is an anaerobic environment, that is very much suited for prebiotic earth, like the earth which was before life appeared. So from here came the theory of biogenesis, that life can only appear from another living or compound, or whenever you from any other source of life, but life cannot appear from non-living matter, which was suggested by Aristotle in spontaneous generation theory. So in this theory, in theory of biogenesis, was this Louis Pasteur proved by this experiment that life cannot appear spontaneously. So he this is a bacteria growth media, and he applied heat over it so that if anything is present inside, it will be killed. Then he let the flask rest for some time, so it was seen that no bacteria is present. And since the neck of the flask is like this, it is very difficult for anything to come inside easily. Again, in another experiment, he heated the flask and then removed the neck, and he saw bacteria was present. So the spores in the air, that those spores entered in the flask and they started growing. Next, when he tilted the flask sideways and also allowed to rest, then also bacteria was present. So it was quite obvious that when the spores came in direct contact with the growth media, then bacteria started multiplying itself, and therefore we can see a bacterial growth. So Reddy also performed this experiment. He kept meat and he opened an open container, and of course there were maggots in the meat, and because flies came and laid their eggs. But when he it was in a cork sealed container, then no formation of maggots. And also when there was a gauze covered container, then it was only that the smell of the meat was there. So the flies came, laid their eggs, but on the gauze itself, the maggots formed. And in this Spallanzani's experiment, he took a meat gravy, he boiled it, and the flask was kept open and was sealed. In this open flask, it was steaming with microorganisms, and in the sealed flask, this gravy was free of any microorganisms. So these experiments proved that you need air to carry the spores of the microorganisms, and if it is so, then only from those spores the new life will be created. It is not possible that the maggots start appearing on their own. Only when there is a contact with the air, when flies can come in and lay their eggs, only then the maggots can form. So these experiments prove that spontaneous generation is not possible. This is known as theory of biogenesis. Next is abiogenesis versus biogenesis. So in the abiogenesis or spontaneous generation theory, it states that life can arise from non-living things. So Oparin, he reasoned that O2 prevents the synthesis of certain organic compounds. Therefore, he assumed that the earth must be anaerobic in nature. So Oparin proposes spontaneous generation of life occur once, but was now impossible because the conditions on the earth has now changed. And he also argued that this primordial soup could be created in an oxygen-less atmosphere, not in a with oxygen atmosphere. So this biogenesis theory states that every living thing came from a pre-existing living thing, which is actually true. So in Reddy performed this, no maggots appeared in meat when flies were prevented from laying eggs. Spallanzani performed that microbes were present in the air and it could be killed by boiling. Louis Pasteur performed the experiments which showed that bacteria do not spontaneously appear in sterile nutrient-rich media. It has to appear by means of spores. So abiogenesis and biogenesis, these are proposed by different persons, and also their concepts are very, very different. So it states that life is originated from non-living compounds, and it states that life originated from pre-existing living forms. Abiogenesis is the theory describing that life originated from inorganic substances, and this from pre-existing living matter. This is not scientifically proved, and this is scientifically proved by experiments. And this is based on observations only. This is based on experiments. So this here suggests that theory of biogenesis is more relevant compared to theory of abiogenesis or spontaneous generation. So next, moving on to the aggregates that I have talked about before, the quasarvates and the microspheres. So this is Miller's experiment where he added water, methane, ammonia, hydrogen, but no oxygen, and then he allowed them to condense. So he used paper chromatography to show that the flask now contains several amino acids as well as other organic molecules. So in his work, years of work involved, he identified that 17 out of the 20 amino acids could be formed in this way. All purines, pyrimidines used in nucleic acid synthesis can be formed like this. But ribose cannot form like this. So nucleotides cannot be formed in the lab like this. But in 2015, they have reported that the 2 nucleotides out of the 4 was used by life as well as glycerol 1-phosphate. So they were able to create that in the lab using all these simple organic molecules. So this mixture was kept circulating and continuously boiling, then condensing the water. So in this way, it was obvious that when whatever the substances that were trapped here, they were actually these substances, amino acids, purines and pyrimidines, etc. Now, what are coacervates? So in this primordial soup theory, we have seen that all these amino acids, proteins, hydrocarbons, they form aggregates when they come together. So these coacervates are actually the amino acids when they spontaneously form droplet like thing or round structures, then we call it coacervates. So these are bead like structures without any membrane, and they are bound together, so they have a distinct internal environment and an external environment. So the primordial soup theory, this provided an ideal medium for the formation of these coacervates. So since primordial ocean was large, water can hold a large number of coacervates for a long period of time without being disturbed. So this amino acids formed under conditions very similar to the Earth's primordial environment, and they form these drops called coacervate drops, which contain a lot of substances like amino acids, etc. inside them. So they are mostly made of aggregated lipids, and they can bud off from each other, and they form a semi-permeable membrane. When they get too big, they divide. So they are similar to microspheres. Now, what are microspheres? Microspheres are small spherical particles, and they are tiny droplet-like structures made of organic protein molecules. And the term microsphere was given by Sidney Fox, and they are free-flowing particles. They are very similar to coacervates. They are also able to absorb things from their surroundings, and they are spherical in shape. They can bud and move, and some people consider them as the first living cells, but they are not living structures because they do not contain any hereditary material like nucleic acids and all that. So they cannot be considered as first cells, but they are obviously very important in the formation of protocell. So what are the similarities? They are set up from the environment by a membrane-like boundary. So they are given a boundary. That means they have a specific inside environment and a specific external environment. So they can take up certain substances from their surroundings, and certain chemical reactions take place more easily inside these structures than they can in water. So coacervates can grow, microspheres can bud. And what are the differences between them? Difference is coacervates are mostly aggregates of lipids, and microspheres are mostly aggregates of amino acids or proteinoids. Coacervates can grow, microspheres can bud from each other, and there is a single membrane and there is a double membrane. Also, coacervates are aggregates of lipids, and they are aggregates of protein compounds. So both the coacervates and microspheres, they are spherical in shape. They are composed of protein molecules inside, and they are organized as a membrane, and they are able to grow and replicate. So all these features make them very similar to cells, but still they are not considered as true live cells, as they do not have any hereditary material to transmit from one generation to the other. So next, coming to the geological time scale. So geological time scale, as you can understand, right from the beginning of the life on earth till now, the entire time scale has been classified like this. So if we consider from the past to coming to the present, we have this broad divisions called era. So the first one was Precambrian, then Paleozoic, Mesozoic, and the most recent one now is known as Cenozoic. So this Precambrian era was 3000 million years ago almost, and therefore it had 3 periods. So this era is divided into periods, period is divided into epoch. 3 periods, upper, middle, lower, and in each of them we can start from the lower period. There were planktons and prokaryotes. In the flora section, the first forms of life were prokaryotes and planktons. In the middle Precambrian period, the appearance of first eukaryotes. And in the upper Precambrian period, the first multicellular organisms came into existence. Next, moving on to the Paleozoic era. This Paleozoic era is divided into these 6 periods: Permian, Carboniferous, Devonian, Silurian, Ordovician, Cambrian. So Cambrian, starting from Cambrian, so you can understand why this era is named as Precambrian, because it was just the era before Cambrian period of Paleozoic era. So therefore this is known as Precambrian. And now coming to the Cambrian period, there are invertebrates. The first invertebrates appeared here, and also the origin of algae. Now in the next period, which is Ordovician, we can see this is the period of invertebrates, because the invertebrates appeared first in the Cambrian period, and in Ordovician period it will start becoming dominant. And also there is appearance of first land plants. So till now only the plant life was only found in the water. Now we have seen that there is appearance of first land plants. Now in the next period, which is Silurian period, we can see earliest fishes and land invertebrates. So now animal world also started developing in the land. So fishes started in the water, and in the land invertebrates started appearing. In the moving on to Devonian period, this early fishes now became dominant, and therefore this is known as the age of fishes. Also progymnosperms started appearing here. In the next period, there is Carboniferous, which is again divided into 2 epochs, Mississippian and Pennsylvanian. Mississippian epochs have seen the earliest reptiles. And the earliest reptiles, then moving on to the Pennsylvania, we have seen here earliest amphibians and abundant echinoderms. So echinoderms have also taken dominance over here. And next in the flora section, we can see the ferns and the bryophytes. So moving on to the next period, which is Permian, there you can see the mammal-like reptiles, or the missing links between these 2 major classes, between reptiles and mammals. And next is the Mesozoic era. So the Mesozoic era, we are all familiar with Jurassic period. So these 3 periods are in the Mesozoic era: Triassic, Jurassic, Cretaceous. In the Triassic, there is not much significant event, and therefore the first dinosaurs appeared in Triassic. And in Jurassic, it is known as the golden age of reptiles, or the rise of the dinosaurs, the dominance of the dinosaurs. And the first birds also appeared in this particular period. And in case of flora, the dicots appeared, the conifers, etc. And in the Cenozoic era, moving on to the Cenozoic era, which is the recent era, and it is divided into 2 major periods, Quaternary and Tertiary. Tertiary period is divided into 5 epochs: Paleocene, Eocene, Oligocene, Miocene, Pliocene. And if you look at these 4 epochs, the mammals and the birds were like in the Tertiary period, the mammals and the birds started becoming dominant. And this is in the flora section. It is known as the age of angiosperms because the dicots were already appeared in the previous period, which is Cretaceous. Now in Pliocene we see the human evolution starts, but the age of human beings becomes moves on to Pleistocene. The Quaternary period is now divided into two epochs: the recent one, which is known as Holocene, and the previous one, which is known as Pleistocene. So in the Pleistocene epoch we see that it is known as the age of human beings. And again in the recent one, which is known as the Holocene, this is the age of mammals. And in flora section we have monocots also. So now the monocotyledons are taking dominance and they are becoming very significant in the flora section. So in this way, starting from 3000 million years ago till now, we have seen how this evolution has taken place. So here also this mass extinction are given over here, like in Ordovician for 85% was death rate, and Devonian again, then Permian, then Triassic. So these are like mass extinction took place in these particular periods. And this is how the evolutionary time scale has progressed through time, and how the starting from the appearance of prokaryotes till today, the age of human beings, whatever the developments have taken place, this is given in this table. And the last thing in this topic is the human evolution, like how human evolution has taken place throughout the years. We already know that we share a common ancestor with gorillas, chimpanzees, bonobos, etc. So this common ancestor in around 9 million years ago, they branched out into gorillas, and this branch, this is a shared ancestor by humans, chimpanzees and bonobos. So from this, again around 6 million years ago, the humans became separated and the chimpanzees and bonobos became separated, and this is known as the genus Pan. And today this genus is known as genus Homo, and from this Australopithecus to Homo habilis to Homo erectus to Homo heidelbergensis, Homo sapiens, today we have this Homo sapiens. So this evolution is mostly on the basis of the adaptations for walking, bipedal locomotion, because earlier they used to walk on 4 and now we walk on 2. And then canine teeth is smaller compared to other carnivores, then enlarged cheek and teeth, jaws, then the muscles of chewing muscles, then slightly larger brain capacity. So brain capacity has played a very important role, because you see in every species the brain capacity or the skull shape is changing quite drastically. So this is a very important factor in human evolution. And from there, smaller jaws, cheek teeth, long legs, long distance walking, running, so these came into existence. And also the sophisticated stone flakes, the tools for hunting, brain size has increased to 1200 cc. So large brain, the last one, Homo sapiens, we can see that the brain is the largest, 1400 cc, the skull capacity, and the small face is small, tucked below the brain case, and this round cranial vault. The language is very proficiency in language, and capacity for art and thought. These things became very crucial for human evolution. So the skull capacity, the locomotion, bipedal locomotion, and the muscles for chewing, the teeth, the jaws, the facial thing, and also the art and thought thing, and the language, these are very crucial for human evolution. And this is how human Homo sapiens has come into existence today, from starting from its very ancestor, which is shared by gorillas, chimpanzees, bonobos, etc. So In the next video, we will move on to the mechanism of evolution, and we will start with how evolution has taken place exactly and what are the different types of evolution. So, thank you.